Good afternoon or good morning.
Hello, hello, hello.
Hello, hello. So it's time to start. Welcome back to the SDGs workshop. Let's start today's session. Today, the first group is UNDIP Group 1. If we are ready, please start your presentation.
Thank you very much, Sensei. Now is representative from UNDIP or Diponegoro University. So could you please, the first group is about the seagrass. Please, team of the seagrass, could you please present? Can you share the screen, the group, Reza?
So let me introduce my team. My team from Diponegoro University, Faculty of Fisheries and Marine Science. So there are four people here. First, there is Hiskia Sitepu, Habib Muhammad Rehan, Faizal Safa, and me, Muhammad Reza Yanur Rahman. So today we will presentation about seagrass, or in Indonesia, well known as a lamon. So seagrass, also known as lamun, is a flowering plant or angiosperma that thrives and grow in shallow water bodies. It possesses roots, rhizome, leaves, flower, and fruits. So, and lamon reproduce by generative and vegetative. Seagrass meadows are extensive area formed by one or more types of seagrass. The seagrass ecosystem is an ecological system comprising biotic and abiotic components within seagrass meadows. And the characteristic or in Indonesia ciri-ciri from the seagrass first is found in a shallow coastal water on muddy or sandy flats. Then occurs at the lower tide level near mangrove, usually near a mangrove forest or in a coral reef. That the seagrass can thrive in calm and sheltered water at depth of up to 30 meters. Highly depend on sunlight penetration into the water. And five, capable of optimizing metabolic process when fully submerged, including generative reproduction, then able to survive in saline environment. And last, possess a well-developed root system. So then lamon, there are 52 species lamon in this world, and 12 of them found in Indonesia. So in Indonesia, there are two classified families. First is Hydroceritaceae and Potamogetoniaceae. And most of seagrass species, there are similar external morphology characteristic by long, thin, ribbon like leaves that have water channel and grow from creeping rhizome.
Yeah. Thank you, my friend Reza. Okay, I will continue the presentation. Enhalus Acorides. The classification of Enhalus Acorides as follow. Divisionnya Anthophyta, class Angiospermae, subclass Monocotyledonae, ordo Helobiae, family Hydroceritaceae, genus Enhalus, species Enhalus Acorides. The characteristic of Enhalus Acorides include one, upgrade plant to structure, two, to the 5 leaves energy from the rhizome, ribbon like a leaf, creeping rhizome, rock with branching, non-branching, and surrounded by a thick outer skin, long and hard and roots. Thalassia emperis. By characteristic of Thalassia emperis, divisionnya Anthropita, kelas Angiosperma, Angiosperms, subclass Monocotyledonae, ordo Halobiae, family Hydrocharacteriaceae, genus Thalassia, species Thalassia empirici. The characteristic of Thalassia empirici leaf are elongated, slightly with some brown lines. Two, rounded and blonde leaf tips, and three, head leaf seed, and four, creeping rhizome. Next. Halopila ovalis. The classification of Halopila ovalis as follows: division Anthopitae, class Angiosperms, subclass Monocotyledonae, order Halobiae, family Hydrocharacteriaceae, genus Halopila, species Halopila ovalis. The characteristics of Halopila ovalis include one, leaf blade oval and straight with clear vein. Two, leaf stalk like ringing from 1 to 4 centimeter. And three, rhizome are creeping and around. Cimodocea rotundata. The classification of Cimodea rotundata as follows: division Anthropita, class Angiosperm, subclass Monocotyledonae, order Halobiae, family Potamogetonacea, genus Cimodocae, species Cimodocea rotundata. The characteristic of Cimodea rotundata include one, slender plants resembling Cimodocea sermulata, and two, strike like rounded to somewhat circular shaped leaves, what the draping of the, and three, rounded leaf tips and hard leaf seed, and four, slim rhizome. Next, Oceana cerulata. The classification of Oceana cerulata as follows: division Anthropita, class Angiosperms, subclass Monocotyledonae, order Halobiae, family Potamogetonacea, genus Oceana, species Oceana cerulata. The classification of Oceana cerulata: one, plain, similar to Cimodocea rotundata, and two, leaves are longer and more rounded. Three, rounded leaf tips with a slight serration. And four, bulky rhizome with short erect shoots, and 2 to 4 leaves on earth segment. Number 6, Holodule uninervis. The classification of Holodule uninervis as loss division Anthophyta, class Angiosperms, subclass Monocotyledonae, order Helobiae, genus Halodule, species Halodule uninervis. The characteristics of Halodule uninervis: 1. Upright plant resembling Holodule pinifolia, and 2. Leaves curved at the tip and are narrow at the base, and 3. Contain small tentacles, and 4. Clearly visible midrib in the center of the leaf, and 5. Creeping rhizomes, and finally the seagrass species Halopila minor. The classification of Halopila minor as follows: division Anthophyta, class Angiosperms, subclass Monocotyledonae, order Helobiae, family Hydrocaritaceae, genus Halopila, species Halopila minor. The characteristics of Halopila minor include: 1. Leaf blades resemble those of Halopila ovalis, but are similar in size. And 2. Fewer leaf veins. And 3. Thin and easily breakable rhizomes. And 4. To exist with seagrass vegetation that does not completely cover the sediment surface. Next.
It can be seen here. This is a map of seagrass meadow condition in 2018. Seagrass bed can be found in almost all part of Indonesia, from North Nias, next Batam, Bintan, Belitung, nextnya Lampung, Makassar, Selayar, Sika, Buton, Wakatobi, Kendari, Ternate, and Biak Numfor.
Benefits. Seagrass has lots of benefits. The benefits of seagrass include primary producer, the habitat of biota, sediment catchers and follow and wave actors, the composition of nutrients, and carbon sinks. Primary producer. Seagrass is primary producer in the food chain in many waters. Seagrass is a primary producer because it has availability of nutrients, phosphate, nitrate, and ammonium. And 2. The habitat of biota. Seagrass is a host that also provides food for marine animals, most of which are small fish. In addition, seagrass also the main food for a number of enriched biota, such as the dugongs and turtles, sediment catchers, and fall and wave arretors. Seagrass has a freeboard and strong rhizome structure that is able to maintain the stability of seafloor sediment and prevent abrasion due to current and sea waves. The composition of nutrients. Another important role of seagrass is to maintain the balance of the marine ecosystem. Seagrass acts as a nutrient recycler and waters where organic matter is returned to the water stock. This composition process with the help of microorganism activity. Carbon sinks. Seagrass is coastal ecosystem capable of strong and absorbing carbon through the process of photosynthesis, which is stored in the form of biomass and stored in roots, rhizome, and leaf so that it can reduce CO2 goes in the air.
Thank you, Hiskia, for this presentation. And I will continue this presentation. Seagrass Biota Association. The community of associated animals in seagrass is divided into several categories. 1. Animals that live on seagrass leaf. 2. Animals that attach to seagrass rhizome. 3. Animals with high mobility that swim beneath or between seagrass leaf. And 4. Animals that live on or within the sediment substrate. Epiphyte. Epiphytes are microalgae that attach to the surface of plants. Epiphyte can contribute to the food chain directly or indirectly. Here, there are several examples of biota association that live in seagrass beds, such as clam or shellfish. There are Tridacna maxima and Anadara. Next, seaweed, example Halimeda, Sargassum, and Padina. There are sea cucumber, example Holoturia atra and Holoturia scabra. There are also seasonal, example Conus tronus, and next jellyfish and octopus. There are sea urchins and reptiles such as sea turtles and sea snakes. There are also several types of fish such as Parupeneus indicus, Spairamia orbicularis, Tylosaurus crocodilus, but there are some biota that are as dangerous as Paracanthurus hepatus, Sinanseia horrida, and Siganus canaliculatus. So, if you want to go to seagrass meadow, you should be careful with this biota. Okay, this is presentation. I will back the moderator.
Okay.
Okay, thank you very much, Group 1. Okay, and now the paper is open for discussion. Are there any question or comment? Okay, I have one question. It is not directly related to today's your talk, but it's from my personal interest. Today you introduced 7 seagrasses.
Yeah.
And all the seagrasses belong to the angiosperm. But the
Yeah.
angiosperms is roughly classified into two groups, so 2 subclasses. So
Yeah.
monocots and dicots.
Yeah.
Okay, but I missed the first classification of the first 2 species, but remaining 5 seagrasses belong to the
Yeah.
monocots. So
Yeah.
my question is why the monocots tend to, so my question is, are there any tendency that the monocots become seagrasses, or they are example of the dicot seagrass? Dicot seagrass. That's my question.
Okay. Thank you, Sensei. First group, can you something explain about the question? Reza? Okay, Sensei. Maybe I can help about the
Yeah.
seagrass.
Okay.
Okay. In Indonesia, as far as I know, there are 9 species, Sensei, that the species distribute in Indonesian seawater. Okay. So they belongs to monocot and dicot. Yeah. But the monocot is very dominant, but depend on the what you call it? Depend on the seawater. Okay, depend on the seawater. So I don't know exactly why. For example, in my area in Semarang, the dominant of the monocot, for example, yeah, because this is very interesting. So now I observe, I research not about the taxonomy, but the function of the seagrass to catch the carbon dioxide from the atmosphere and then go to the seawater through the diffusion, for example. And then they will catch by the seagrass. Okay, but Sensei, in Indonesia, is not all of the what you call it the coastal area can grow good for the seagrass. Why? Depend on the area. Depend on the area. Sometimes the fishery destroy of the area, something like that. But as far I know, there are 1 or 2 researchers from Indonesia. Uh, they make the what you call it the initial experiment how to grow, how to plant the seagrass. This is in Celebes area, the South Celebes Island. So the growing of the seagrass is very good, but it need budget, Sensei. So the problem is this: if your student want to study about this seagrass, especially related with your question, it will better. Yeah, because something like Sato Sensei, bring the student go to the Karimunjawa Island. Yeah, on this week. Yeah, it's very interesting to learn to study about seagrass. Like this, Sensei. Sorry if not so exactly, but this is the overview of the seagrass in Indonesia. Thank you.
Okay, thank you very much. And are there any question or comment?
I have a question.
Please.
Yeah. Are these creatures found only in Indonesia?
Yeah. Yeah.
You mean
yeah.
that this is a specific in Indonesia?
Yes, this is the species in Indonesia. So as I mentioned to you, or I explained to you, as far I know, there are 9 species in Indonesia. But the team or the first group have already present about 7 species. Okay, so not all of the Indonesian seawater can find all of the species. Sometimes only 1, 2, 3, or something like that. But if we total, as far I know, is 9 species in Indonesia. Thank you.
Thank you.
Okay, thank you. So are there any question or comment? Okay, if no, let's move to the next session. Thank you very much, Group 1.
I have one question.
Welcome.
Okay. Please.
Please.
Sorry. How does protecting seagrass relate to the SDG?
So the protecting of seagrass related with the SDG is to protect or to absorb of the carbon dioxide. So now the government stimulate many organizations in Indonesia to to stimulate the plan of the seagrass in some area, but not yet successful. Why? It need the first is budget, the first, and the second is we need organization with full support manpower, something like the student. Okay, so there are many the thing that affected of the program, but we have already done something like my research now. I advise some master student to learn about the seagrass, how to absorb the carbon dioxide from the atmosphere, something like that.
The answer is enough.
Do you have still question?
No, that's all. Thank you.
Okay, arigatou gozaimashita.
Okay, are there any question or comment? Okay, if no, let's move to the next session. Thank you very much, Group 1. And
Welcome,
next,
Sensei.
yeah, thank you very much. And next group presenter is UNDIP Group 2. So if we are ready, please start your presentation.
Okay, good afternoon, everyone. My name is Adli, and I'm from Diponegoro University. Today, me and my friend will present about the benefits of seaweeds. Algae, or we know as a seaweed, is a broad term used to describe aquatic eukaryotic organism that can undergo photosynthesis, but lack the vascular system and structures such as leaf, stems, and root they are found in plants. Seaweeds are generally anchored to the sea bottom or the other solid structures. Seaweed can provide essential nursery for fisheries and other marine species. Seaweeds often dense growth on rocky shores or accumulation in shallow water. The water depth of seaweeds about 50 meters or less. By the pigment compound, type of seaweeds divided into three: Rhodophyta or red seaweed, Phaeophyta brown seaweed, and Chlorophyta green seaweed. By the size, the seaweeds divided into two: macroalga and microalga. The nutrition of seaweed. Seaweed have been consumed as a food and medicine for centuries. Seaweeds have high concentration of vitamins of the nutrients. Seaweeds commonly referred to superfood. Seaweeds contain chlorophyll, which is one of the reasons why they can be so beneficial to our health. Like plants, they use chlorophyll to trap the sunlight necessary for them to undergo photosynthesis, which is why chlorophyll is considered a chelate. During the photosynthesis, the energy absorbed by chlorophyll transforms carbon dioxide and water into carbohydrates and oxygen. A chelate is a chemical compound composed of a metal ion and chelating agent that plays a vital role in oxygen transport and photosynthesis.
Good afternoon, everybody, and I will continue the presentation. So in general, seaweed nutrition is as follows. Protein and amino acids up to 60% protein by dry weight, vitamin A or beta-carotene, C, ascorbic acid, E and K, many of the B complex vitamins including B1 or thiamine, B2 or riboflavin, B6 or pyridoxine, choline, biotin, folic acid, pantothenic acid, and B12 or cobalamin. Minerals and trace minerals including iodine, calcium, chloride, chromium, copper, iron, magnesium, manganese, potassium, phosphorus, sodium, and zinc. Omega-3 fatty acids including EPA, DHA, GLA, and ALA, active enzymes, and phytochemicals like chlorophyll, fucoxanthin, and other plant pigment. Next. And these are the examples of seaweed products. There are hijiki, wakame, ogo, kelp, nori, kombu, arame, dulse, simos, spirulina, blue spirulina, and bladderwrack. And about many microalgae species grow in turbulent waters, can produce hydrocolloid compounds that provide the necessary flexibility and elasticity to prevent breaking in the rough currents. These hydrocolloids, including agar, sodium alginate, and carrageenan, are utilized extensively in the food industry for their gelling and thickening capabilities. And there is seaweed potential as biofuel. Seaweed efficiently use CO2 and are responsible for more than 40% of the global carbon fixation, with the majority of this productivity coming from marine macroalgae. Algae can produce biomass very rapidly. All algae have the capacity to produce energy-rich oil, and a number of microalgal species have been found to naturally accumulate high oil levels in total dry biomass. Oil content of microalgae is generally found to be very high, which exceed up to 80% by weight of its dry biomass. The algae can be converted into various types of renewable biofuels, including bioethanol, biodiesel, biogas, and photobiologically produced biohydrogen. About 5,000 to 15,000 gal of biodiesel can be produced from algal biomass per acre per year, which reflects its potentiality. There are several advantages of algal biomass for biofuels production. The first is the ability to grow throughout the year. Therefore, algal oil productivity is higher in comparison to the conventional oilseed crops. And second is higher tolerance to high carbon dioxide content. The consumption rate of water is very less in algae cultivation. No requirement of herbicides or pesticides in algal cultivation. The growth potential of algal species is very high in comparison to others. Different sources of wastewater containing nutrients like nitrogen and phosphorus can be utilized for algal cultivation apart from providing any additional nutrient, and the ability to grow under harsh conditions like saline, brackish water, coastal seawater, which does not affect any conventional agriculture. Next, then there is potential algal for mitigation. Because of algal importance in marine ecologies and for absorbing carbon dioxide, recent attention has been on cultivating seaweeds as a potential climate change mitigation strategy for biosequestration of carbon dioxide, alongside other benefits like nutrient pollution reduction, increased habitat for coastal aquatic species, and reducing local ocean acidification. Algae cultivation in the open ocean can act as a form of carbon sequestration to mitigate climate change. Studies have reported that nearshore algae forests constitute a source of blue carbon, as algae detritus is carried into the middle and deep ocean, thereby sequestering carbon. Thank you.
Okay, thank you very much. Interesting talk. Now the paper is open for discussion. Are there any question or comment?
I have a question.
Oh, please.
Hijiki and wakame are also eaten in Indonesia.
Okay, so I think he is asking about so hijiki and wakame is very popular as a food in Japan. So
Yes.
he is asking that
No.
in Indonesia it is a popular food.
Says wakame, taberobono wa. So wakame as the food thing, yes, you can find in Indonesia. But wakame as seaweed, which live in seawater, no, we don't have. Yeah, we don't have. We have something like wakame, we called sargassum. Okay, but it's very small. And also, this is the challenge from your side, from the Kwangsei Gakuin University student who want to study about seaweed in Indonesia. Is very, very big challenge because we just know only 1 or 2 or 3 species which is very, very important thing to export overseas. So not look like in Japan, wakame, hijiki, odonori, or something like that. No, we don't have many. Even if you go to the coastal area, you can find many, many species, but only 1 or 2 or 3 species that we process and then export to overseas. They are Equema spinosum, they are Capapicus alvarezi, and also Gracilaria. Okay, but something like Sargassum is only take from the natural coastal area. So that's mean not so big cultivation. Yeah. Even we have many, many species, Sensei. Thank you very much.
Okay.
Thank you.
Okay, I have one question. So today, the group to introduce many various aspects of seaweed, how to use it. So biofuels or nutrients or something like that. But each country or each tribe has its own traditional medicine called ethno medicine. And I think in Indonesia, you have jamu. So my question is, are there any formula of jamu which include seaweed?
Hello, hello Aldi Group. Can you explain about the jamu from seaweed?
As far I know, there's no jamu from the seaweed in Indonesia.
So are there any reason why seaweed is not used for jamu?
Sorry, Sensei. I will give the information. Okay.
Yeah.
So in Jepara is near from Semarang. It's also the seawater area which Sato Sensei will visit there. Okay. There is one species we called kaulerpa. Yeah, we got kaulerpa, and local name is latoh, and English name is sea crab. Okay, so there is special cafe, there is special resto with sell the latoh, the sea crab or kaulerpa as food. As in say as food, but not yet as jamu. Not yet as jamu, but as food, yes. I usually eat the kaulerpa when I went to the Jepara. And the function for human body is fresh. So if I have already eat, for example, one, what you call it, 1 piece or 2 piece, something like that. So the body is fresh. I don't know exactly why. So if you ask me why there is no jamu from seaweed, so this is the big problem in Indonesia, Sensei. Yeah, there is no study program of phycology. There is no yet study program of phycology. Not something like in Japan, Hokkaido University or Kagoshima University. There is special program, yeah, study program. So that means there is not yet the student can study special for phycology and undergraduate program. Not yet, Sensei. So this is our challenge. So that's why if something like Kagoshima University have already collaboration with Diponegoro University. If you can send the student and explain about the seaweed, wow, it's very, very fantastic, Sensei. Thank you very much.
Okay, thank you very much. Okay, are there any question or comment?
There is no hijiki. There is no hijiki. Hijiki is special in Japan.
Okay, if no, let's move to the next presentation. Thank you very much, Group 2.
Arigato sensei, arigato,
Okay,
arigato
terima
gozaimashita.
Okay, next presentation is given by KGU Group 2. So if we are ready, please start your presentation.
Group 2、Biodiversity を、さらいけ、National and its Maintenance、ユースケ、東、翔太、牛岡、一木、山本、伊織、吉本。関西学院ユニバーシティ、contribute to the achievement of the Sustainable Development Goals, the 17 Goals for Global Change. Through education, research, and management while practicing the school motto, nurturing creative and competent global citizens who embody mastery for service. By transforming society with compassion and integrity, our science school is located at Sanda campus in Sanda City, and Sanda is promoting nature conservation activities. In this respect, the 15th goal the SDGs, let's protect the abundance of land goes with Sanda City's activities. Sanda City is home for precious Saraike. Marshland are placed with many rare and precious landform flora and fauna that are rare even in Japan. We call this kind of landform Satoyama. Satoyama is mountainous ecosystem that exists as a result of human intervention in the forest near village in Indonesia. Satoyama also exists and Gathering Hariman-Sarak National Park is one of them. This means that Satoyama will be lost without human intervention. In Sanda, we are working to protect Satoyama. We now present what we have experienced and learned from our participation in these activities.
The Saraike Marsh we visited has a diverse array of flora and fauna. Here we would like to introduce some of the most impressive organisms. First, iron bacteria. These are bacteria that oxidize water-soluble divalent iron ions and are widely found in soil. A film similar to that of oil is produced on the surface of water when ferric hydroxide is generated. The distinguishing feature is that it does not return to its original state when cracked. Second, Yamadori mainspring. This is a large fern that grows in wetland. There is a theory that it is a survivor of the Ice Age colonies. In Japan, it has shifted its habitat to the lowland over time. Saraike marshland is not a lowland habitat, but it does occur there. Third, Setouchi salamander. We discovered salamander's eggs during our first visit and got knowledge that the eggs had hatched during our second visit. Salamander is classified as an endangered species. To protect Setochi salamander eggs, a net was placed over the area where eggs were located. Therefore, the eggs would not be crushed by animals such as boars. Fourth, Kawabata morocco. This variable fish is designated as a domestic rare wild animal and plant species. In Saraike marshland, it lives only in Kamisaraike pond. Predation by black bass, a non-native species, has caused the extinction of the Kawabata morocco that lived outside of Kamisaraike pond. Fifth, Hacho dragonfly. This is one of the world's smallest dragonflies. The body color is red for males and light yellow for females. We are able to observe the female. These dragonflies are one of the main organisms in the marshland, and their presence is a sign that the marshland is well maintained. Lastly, agate flower. It is one of orchid families that inhabit the marsh. In Saraike Marsh, it blooms in August. It is named agate flower because its flowers look like a white agate spreading its wings.
There are three main challenges to maintain wetland biodiversity. The first is the forestation of wetlands and the growth of large grasses. The Saraike wetland consists of lowland forest, wet forest, reservoir, and grassland. If trees in a forest are less than cut, they will grow larger and become densely populated. If the forest grows too much, sunlight will not reach the forest, making it impossible for ground cover plants to survive. Similarly, tall plants dominate grassland and the like, leaving no place for other plants to survive. In addition, wetland forests such as cherry alder trees are necessary for the wetland ecosystem because these trees serve as spawning and habitat areas for animals. A second challenge is survival of marsh. Saraike marsh has various sizes. When the soil collapses due to heavy rains or floods, waterways form between the wetlands. If the wetlands are connected by a waterway, all the water in the wetland will flow out. Water runoff and evaporation can reduce marsh areas. Trees tend to disappear. Finally, the third challenge is invasive species. Saraike marsh is home for various of rare species, but they are in danger of extinction due to invasive species. Raccoons, wild boars, and black bass are examples of invasive species that have appeared in Saraike marsh. Invasive species feed on native species and destroy habitats of native species. In fact, many native species that used to live in the Saraike marsh but have become extinct due to invasive species. In order to protect the rare species of Saraike marsh, it is necessary to prevent invasion of invasive species. Human management is necessary to maintain the biodiversity of the Saraike marsh.
Now, let us conclude our presentation. In order to solve these challenges, various conservation strategies have been taken by Sanda City, its residents, professional organization, and volunteers for the Saraike wetland. We would like to share 3 strategies with you. The first strategy is the artificial deposition of the plant's transmission. By cutting down tall trees that grow in the wetland, sunlight can reach the ground and other plants are able to leave nutrients for growth. By cutting down tall trees, the wetland can keep the conditions for growing the rare plants. Similarly, the community used to remove bamboo and large grass in order to keep sunshine on the ground. The second strategy is to make water flow through the swamp. Look at this picture. In this way, the water is stuck in the wetland by peeling stones. In the neighborhood, many pipes are stuck on the ground and are used for survey of the water volume. So saving water is essential to preserve the environment of the natural rich wetland. The third strategy is installations of electric fences and the registration of natural monuments in the ecosystem. As shown in the following pictures, an electric fence is installed in the area which has their plant to prevent wild boars from wild boars from destroying these plants. This is because wild boars dig the soil and cause the plants to die by tramming the soil. In addition, the ecosystem is registered as a natural monument of the prefecture. It is prohibited to enter the prefecture except for those concerned. So that is can prevent invention of black bass by acknowledging the importance of the people in the prefecture. These strategies protect the ecology and keep biodiversity in Sarake wetlands. In Satoyama, the community manages nature by adding artificial controls. However, if no one controls the conditions of the wetlands, it will change into a big forest, and the rare plants will disappear from there. Therefore, sustainable activities are required for the maintenance of the wetlands. In Japan, we are promoting the Satoyama Initiative, which aims to realize sustainable use of natural resources by using methods and data established through activities in Satoyama. In many parts of the world, the people conduct case studies like we introduced in our presentation. We believe that the conversation activities of Satoyama can be used as a reference for overseas conversation activities. Thank you for your attention.
Okay, thank you very much, groups of KGU. Before starting the discussion, I have one comment. So you mentioned about the invasive species and the danger of the invasive species. But one of the examples of the invasive species, you showed the wild boar. But the wild boar is a native species in Japan. So the description is wrong. So it's my comment. So now the paper is open for discussion. Are there any question or comment?
Hi.
Please.
I have one question.
Please.
Just now you mentioned about the animals that are the risk of extinction. So my question is there any strategies done to conserve them?
Sorry, once more time, please.
My question is, just now you mentioned about the animals that are at risk of extinction. So, is there any strategies done to conserve them? Should I simplify the question?
In my idea, people almost in the prefecture or governments need to usually Sarake wetland is locked, so the general people can't enter the place. So it is one kind of protections and strategy for protecting Sarake wetland. Does my answer make sense?
So, is that answer enough for your question?
Yes, yes. Thank you.
Oh, thank you.
I have question. I have
Oh, please,
question.
please.
Sensei, this is very, very interesting in Japan, okay, about the biodiversity in Saitama. So can you predict around 25 years later, how is the biodiversity in your area related with what you call it, increase of carbon dioxide? So as far I know, in Japan is very large, yeah, because the industry country. What do you think about that? You understand what I mean?
I think it will be decreasing, maybe. Yeah, because of our our main energy resources will be changed in the future, maybe.
That's why.
So the Saraike Shitsugen, the problem of Saraike Shitsugen is not directly related to carbon dioxide problem, but the approach in the Saraike Shitsugen may be a good provide a good example how to treat the carbon dioxide, because the in order to maintain the biodiversity in Saraike Shitsugen, many people to enter into the Shitsugen Saraike wetland, and so cut down the tree and try several operations. It means that if we leave the nature as it is, we cannot maintain the biodiversity. The human hand is required to maintain the biodiversity. So I think the carbon dioxide problem is the same. So the older human beings try to reduce. There's such an effort to reduce the carbon dioxide is required.
Thank you, Sensei. So because what will you call it? What will be happen in Japan is our guide, okay, for the future. Yeah, because the industry is very, very high in Japan. You understand what I mean? So when the industry is very high, so they can they will produce much of the carbon dioxide. So, but the Saitama as the presentation is very, very good for the diversity. But 25 later, I don't know exactly. But if you have already explained, it's very, very nice, Sensei. Thank you very much for the answer.
Okay. So are there any question or comment? Okay, thank you very much, Abe-san Sensei. And if no,
terima kasih.
okay, if no,
Sama-sama.
Wait a minute. Next is okay. So let's move to the next session. Next presentation will be given by UiTM Group 3. So please prepare the presentation.
Assalamualaikum and good evening to everyone. We are group number 3 from University Technology Mara. Today we are going to present the topic of molecular conservation. Next. Before we start, let's introduce our members first. My name is Emilia Syahada binti Muhammad Nisam.
And I am Brittany Anak Bujang.
I am Anissa Darwina binti Rosli.
I am Nur Hazika binti Abdul Rahman.
And my name is Nasisha Nelly binti Sharbudi.
So, start with the introduction. For our presentation, we choose Sumatran rhinoceros as our subject for molecular conservation topic. The Sumatran rhinoceros is a distinct and ancient species roam the dense rainforests of Southeast Asia in significant numbers. This species, recognizable by their shaggy appearance, and 2 horns, and this rhino hold a critical ecological role as a primary seed dispersers for a substantial number of plant species, which is benefiting at least 79 different plants. As this rhino roams through the dense rainforest of their native habitats, they play an indispensable role in maintaining the health and diversity of their ecosystems. However, over the years, the Sumatran rhino population in Malaysia experienced a sharp decline due to a combination of factors. Next. Such as habitat loss resulting from deforestation, driven by the agricultural expansions and logging, significantly reduced the substitutable habitats available to these rhinos. Additionally, the species face the threat of poaching, driven by the illegal trade in their horn and other body parts. And also, the population decline was also compounded by the species' low reproductive rates. The Sumatran rhinos have relatively long gestation periods and give birth to only one calf at a time, with several years between birth. This low reproductive rate made it challenging for the population to recover from losses. And according to the International Union for Conservation of Nature, IUCN 2020, the Sumatran rhino is now listed as the critically endangered species, which is highlighting the urgent needs for comprehensive conservation efforts to prevent its extinction. Next.
Okay, hello everyone. Now we will look at techniques to conserve the Sumatran rhinoceros. I'll be presenting one of the assisted reproduction techniques to conserve our Sumatran rhinoceros. Note that assisted reproduction techniques consist of in vitro fertilization, cryopreservation, and frozen embryo transfer. To begin with, I will present the in vitro fertilization (IVF). In IVF, the steps involved are eggs extraction from female rhinoceros, fertilized by sperm from male rhinos in the lab, leading to the development of embryos. These embryos will be transferred to surrogate mothers for offspring development. Next. Now, we are going to look how IVF is conducted on rhinoceros. First, the female rhino was put under general anesthesia so that the oocytes could be extracted from its ovaries using an ultrasound-guided probe. The scientists had developed a special probe to extract eggs from the rhino's ovaries, which were previously unreachable using ordinary equipment. After incubating and developing the egg cells, they were fertilized with top sperm from the male rhino using a technique known as intracytoplasmic sperm injection (ICSI). Few oocytes mature into viable embryos, which are frozen in liquid nitrogen. Jerry, a novel benchtop incubator with integrated continuous embryo monitoring capabilities, are designed to provide an individualized and undisturbed incubation environment. This equipment was used to help with the embryo growth. Next. Another critical step in IVF is the transfer of embryos to the surrogate mother. For this step, there will be selection and sterilization of the rhino bull. This process was conducted by sterilizing the animal with a minimally invasive non-surgical process and cutting-edge equipment. As a sterilized bull, she will consistently signal the reproductive cycle of potential surrogate moms through his behaviors without the risk of impregnating them. This information is very essential for transferring embryos to surrogate moms at the appropriate time in their reproductive cycle. Next. The Malaysian government had shown several efforts in conserving the Sumatran rhinos. One of them was by fertilizing Tam, the last male rhino, with Iman, a female rhino. However, Tam's sperm was of low quality, while Iman at that time suffered from a uterine tumor. At that moment, Iman is considered quite healthy, and though she has a tumor in her uterus, she was still producing oocytes. But there was a probability that the rhino might stop ovulating. The result of the IVF was the fertilized cell degenerated within days after the thaw-out sperm. It was learned that the egg cell had failed to divide after fertilization, thus no embryo was formed. This conservation effort was considered failed, but the process of retrieving eggs from Iman continued, which was conducted by a team of experts from the Leibniz Institute of Zoo and Wildlife Research in Berlin, Germany. This conservation efforts takes place in the Tabin Wildlife Reserve in Dahat Datu together with the Borneo Rhino Alliance in Malaysia. Since Tam, the last male rhino has died, the Malaysian government tried to obtain sperm from rhinos in captivity in Indonesia. This led to collaboration between Malaysia and Indonesian governments to carry out IVF of Sumatra rhinos. In this process, Iman will be put under general anesthesia for egg extraction, but this is very dangerous because Iman has large fibroids in the uterus, could be at risk as the blood vessel might puncture if she moves suddenly during the egg extraction process. After the successful collection, the eggs had to be taken in a buffer solution kept at the rhino's body temperature to where the IVF would be conducted. Later, a high-quality male sperm will be chosen. If this IVF process results in an embryo, it will be implemented in a surrogate mother. This is the very first attempt at IVF using eggs from an aging and sick female with the sperm of a fit male. Therefore, the chances of getting offspring at first attempt are almost close to zero, but a process of refining techniques and protocols after several attempts may provide success in conserving the Sumatran rhinos. That's all for me. Now I'll pass to another presenter for the next techniques.
So for the next technique, which is cryopreservation. So what is cryopreservation? Cryopreservation is used to preserve biological material at a very low temperature. And this is in order to slow the biological process and preserve the viability of the preserved cells or tissue. And this is one of the ex situ conservation method for a long-term preservation. And there's a few examples of cryopreservation, such as embryo cryopreservation and semen cryopreservation. Next. So how does the process happen? So for the semen cryopreservation, the male rhino was put under anesthesia, and then the sperm is collected from the male rhino by using a process called electroejaculation. And this involves with gentle electrical stimulation to induce the ejaculation, and then the semen is mixed with a special solution that protect the sperm cells during the freezing. This solution also help prevent ice crystal formation, and the prepared semen is frozen using a controlled freezing process, slowly bringing down its temperature to preserve the sperm. The frozen semen is stored in a liquid nitrogen at extremely low temperature, and this preserves sperm viability for extended period. When needed, the frozen semen can be thawed and used for artificial insemination. This involves in introducing the thawed sperm into the female Sumatran rhino reproductive tract to achieve fertilization. And how does it help with conservation? It helps with the semen cryopreservation can be used in assisted reproduction techniques such as IVF and artificial insemination. And cryopreservation allows genetic material to be stored for extended period. Next. So how the embryo cryopreservation process happen? So the embryo development through in vitro fertilizations. Then the fertilized eggs develop into embryo in the lab for a few days, and to prevent damage from freezing, the embryos are treated with a special solution that protect their cells during the freezing process. And the embryos are carefully frozen using a process called vitrification, where they are rapidly cooled to a very low temperature. And this prevents the formation of ice crystals that could damage the cells. And the frozen embryos are stored in cryogenic storage tanks, which keep them at an extremely low temperature for a long-term preservation. When needed, the stored embryos can be towed and transferred into the uterus of female Sumatran rhino. If successful, the embryo could develop into a calf. And how does it help with the conservation? Cryopreserved embryos can be towed and transferred into surrogate mother. And cryopreservation also provide opportunity for future advancements in reproductive technology or genetic engineering. I will pass to the next presenter.
The next molecular technique for conserve the Sumatran rhinoceros is frozen embryo transfer, an artificial method of breeding that uses frozen fertilized eggs, also called embryo. Next. The frozen embryo transfer are an artificial method of breeding to an animal where pre-implantation embryo are removed from a female animal and transferred to the reproductive tract of another female of the same species in which they develop to maturity. Moreover, the use of frozen embryos, there is only one retrieval that can cover multiple cycles. Compared to fresh embryo transfer, a repeated fresh embryo transfer requires repeat egg retrievals, which can lead to increase the risk of negative side effect, which is stimulation of the ovaries. However, being much cheaper, the process like egg harvesting, insemination, sorry, development of embryo will die completely, then the embryo will be freeze. After years, a frozen egg will thaw and transfer into endometrium of the animal. Next. As cryopreservation, the embryo being frozen to -30 to -30 degrees Celsius under the liquid nitrogen. Then how the frozen embryo will thaw. The embryo that in vitro frozen embryo will thaw rapidly in order to prevent any injuries from recrystallization. So the use of glycerol as chiroprotectant applied in order to have high survival rate by the fast cooling rapid thawing. Then the embryo and the chiroprotectant with a gentle step, like a stepwise method, was added and removed from frozen thaw embryo to avoid the shock of chiroprotectant. Also, the uses of sucrose is to simplify the frozen embryos. Then the glycerol and sucrose is called a one-step straw method. Then by using a sterile microscope, the cryopreservation treatment of embryo prior to freezing and after thaw was done in the laboratory. Then in vivo, transferred to the recipient womb, which is the endometrium. Next presenter.
Okay, so I am going to talk about SNPs as a molecular marker for Sumatran rhinoceros conservation. Single nucleotide polymorphisms, also known as SNPs, is a genetic variation that develops when a single nucleotide in the DNA sequence is changed. SNPs are widely used in genetics and genomics research. They are used for various purposes, including mapping disease genes, understanding genetic diversity, population genetics, forensic analysis, and evolutionary study. Next. There are a few steps to obtain SNPs. Step 1 is sample collection. Samples such as blood, hair, or tissue were collected. Non-invasive methods are preferable to minimize the disturbance. Step 2 is DNA extraction. High-quality DNA was extracted from the samples collected. DNA quality is crucial for accurate SNP identification. Step 3 is sequencing strategy. Sequencing approach was decided whether to use whole genome sequencing for comprehensive SNP information or targeted sequencing to focus on specific genomic regions. Step 4 is data analysis. Raw sequencing data was processed and cleaned to ensure accuracy. Align the sequences to a reference genome if available. Step 5 is variant calling. SNPs were identified by using specialized bioinformatic tools. Step 6, quality control and filtering. Quality control measures were applied to remove sequencing errors, low quality variants, and false positives. Step 7 is annotations and functional analysis. Annotate the identified SNPs to understand their potential effects on genes, proteins, or regulatory regions. Lastly, step 8, population genetics analysis. SNP data was analyzed to assess genetic diversity, population structure, relatedness among individuals, and effective population size. Next. There are a few advantages of using SNPs. For example, SNPs are used for genetic diversity assessment. SNPs are excellent markers for assessing the genetic diversity within a population. By studying the genetic makeup of individual rhinos, conservationists can understand the extent of genetic variation present. The second one is SNPs are used for conservation breeding. In captive breeding programs, SNPs can be used to manage the genetic diversity of the captive population. By analyzing the genetic profiles of individuals, conservationists can make informed decisions about selecting breeding pairs to maximize the genetic diversity and avoid inbreeding. Lastly, SNPs are involved in adaptive genetic variation. SNPs can reveal genetic variations related to specific traits or adaptations that are crucial for the species survival. Understanding these adaptations can inform conservation strategies and habitat management efforts. Next, to verify that SNPs can be used for Sumatran rhino conservation, a study was done in 2021. The study investigates the genetic diversity, population structure, and demographic history of the Sumatran rhinoceros populations using genomic data. The researchers used population structure analysis, including phylogenetic tree and principal component analysis. They examine the extent of private mutational load within each population and whether signatures of positive selection potentially associated with local adaptation differ among the populations. The study suggests that assisted gene flow among populations could mitigate the risk of outbreeding repression and help to conserve the genetic diversity of the species. The study also acknowledged some limitation of the methods used and suggests that future studies should combine genomic data with ecological and environmental data to provide a more comprehensive understanding of the factors that contribute to the decline of the species. That is all for me. I am going to pass the presentation to the next presenter.
Now, I'm going to explain the last molecular technique, which is the induced pluripotent stem cell of Sumatran rhino. This method is newly designed and still in research until today. Scientists have found potential methods to repopulate endangered species, especially the Sumatran rhino. This method is established as the last resort to repopulate a species when the methods involving a living species have failed. Induced pluripotent stem cell or iPSC method uses molecular and reprogramming techniques to convert fibroblasts, which are the connective tissues, into germ cells, which are the precursor of reproductive gametes. To ensure the success of this new method, viable cellular material must be available, such as skin cells or other tissue samples. iPSCs can divide indefinitely in culture and are capable of differentiation into any cell type in the body, including gametes. And once these reproductive gametes are mature enough to form a zygote, it will be implanted into a living species of rhino or any closely related species such as a horse or maybe a bull. Next slide. Let's take a look at how this technique works. Keep in mind that this technique is very complicated, so I have simplified the mechanism and highlight the crucial parts so that we can have an idea how this works. The first step is obtaining the fibroblast and the iPSC culture. The cell lines were obtained from the skin biopsies of the Sumatran rhino, which still have high level of genetic elements. The fibroblast cells lines were cultured at 37 degrees Celsius in the specified medium and supplemented with growth factors. The growth factors are supplemented to ensure the cells are getting enough nutrients to maintain their stability and quality, and so they can grow well. Second, deprogramming and the optimization of the cells. The cells were grown and were induced with a special substance. The cell then were plated on the specialized supplement medium and optimization of the protocol was done. And third, specialized cells tests. Many tests such as spontaneous differentiation, immunocytochemistry, karyotyping, and cardiac differentiation were done to the reprogrammed cell to select the best culture and to test their readiness for further progresses. Next slide. Fourth, RNA extraction and primer design. Cell pellets from the previous extraction were obtained and the gene of interest were identified. This step was performed to design primer pairs for further processes. And the last step is the quantitative reverse transcriptase polymerase chain reaction. Cell amplification was obtained and the only cell that shows high yield will be used. Next slide. For the result, as for now, it was reported that the first step of induced pluripotent stem cells was successful. However, the result of the experiments are far from producing a whole new animal. Further studies are still being held with the help of this successful finding for the future. As one of the molecular biologists that involved in this work, Muhammad Luqman Maisal said, if everything is functioning, works well, and everybody supports us, it is not impossible. Next slide. In conclusion, the extinction of Sumatran rhino in Malaysia is something serious that needs to be paid attention to. Therefore, the Malaysian and other scientists are giving their best efforts to help repopulate the species. As being explained earlier, the 5 molecular techniques, whether has been successfully implemented or still in research, are the efforts to help repopulate the Sumatran rhino. I believe that is all from us. Thank you.
Okay, thank you very much. Interesting talk. So now the paper is open for discussion. Are there any question or comment?
I have one question.
Oh, please.
Can you explain why captive breeding through ex situ cannot conserve the rhino?
I will answer the question. So the question is why captive breeding cannot conserve the rhino. This is because the rhino the pregnancy duration of the rhino takes a long time, which is up to 18 months. So in the meantime, the rhino ages and might get sick or decline in their health. So the offspring produced will not survive long. Other than that, it was viewed as a failure because the lack of living rhino itself, due to the poaching of rhino parts that have killed them. I hope that's answered the question.
Thank you.
Okay, are there any question or comment?
I have a
I
question.
would like to ask a question.
Please.
Sorry.
Okay. So my question is, what are the disadvantages of the old methods, which are IVF and cryopreservation, that will lead to the newest method other than extinction of the female rhinos? Thank you.
Okay, thank you for the question. I would like to answer this question in behalf of my group. So the question is the disadvantages of the IVF and cryopreservation that leads to the newest methods. The answer, to my knowledge, I will answer this question with because IVF or cryopreservation, both of these technologies use semen and ovum of the rhino, and as we know, the long-term freezing can affect the stability of the genes and the outcome, which will cause the cells to lack quality and then result in a failed attempt of breeding it. So that is the disadvantages of these assisted reproduction technologies that lead to the latest molecular techniques. So that's all from me.
Okay, thank you.
Okay, KGU, please.
Are there other endangered animals in Sumatra?
So I'm sorry, he is absent yesterday, so he didn't had yesterday's presentation. So yesterday, the tiger and the elephant it talked about the example of the endangered species. So I have a question. So you mentioned about the SNPs. And by using the SNPs analysis, it was told that there adaptive traits can be detected. So are there any examples of the adaptive traits related to the SNPs?
As to my limited knowledge, adaptable example of adaptable. I think maybe it's to adapt with the environment that they will do the conservation. Maybe I think that is my answer.
And is it so I have a question about the calculation method to detect the adaptive traits. So is it a method related similar to the GWAS? GWAS, do you know GWAS?
No.
Okay. When the genomic sequence is obtained, the disease and the synapse relationship is detected by a statistical approach called the GWAS. But anyway, it's okay.
Okay, thank you.
Yeah. Okay. Are there any question or comment?
Yes, I have.
Oh, please.
And so a while ago, and Malaysian rhino is already extinct in the Malaysia, but is it still alive rhinoceros in Malaysia, Sumatra? Is it still alive?
I will answer the question. So for the Sumatran rhinoceros, in Malaysia, it was extinct, but there's still there's still a population in another country. For now, it's been identified at Indonesia. So for the population, it was estimate around 30 only. Thank you.
Thank you.
Okay, are there any question or comment? Okay, if no, let's take a break. And let's take a break of the 15 minutes. So please come back here 3-5. Okay? Okay, let's take a break.
Okay, thank you very much, Sensei. First of all, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Thank you for the opportunity to present our discussion. So today, we will present the topics about ex situ conservation of Rafflesia, endangered emblematic parasitic species from Indonesia. Okay. Please let me introduce our member first. Next. Okay, so I am Aditya Marcel Junior as the moderator, and then here our team is Raffi Amar Maulana and Saleh Mubarak, Rohma Shifwatul Jinan, Tasya Janwa Shela, Reza Tranadia Felani, Lilia Elizar, and Angelin Irene Christmas. Next. Okay, for the first topic, I will pass to Rohma. Please welcome.
Okay, one of the biggest flowers in the world, which smells like rotting flesh, has been found in the Indonesian rainforest. Rafflesia was discovered in Sumatra by an exploration party visiting Shelley Stamford Raffles and his wife, Lady Sophia Raffles. On the Mana River on either 20 May 1818. The flower, which belongs to the Rafflesia genus, measures over 3.5 feet across and blooms for only 1 week. It smells like a rotten flesh in order to attract flies and carrion beetles. Flower that smells like a rotten flesh in order to attract flies and carrion beetles. They have no leaves, roots, stems, or leaves, growing as a parasite or tetrastigma. They derive all their nutrition from the host. They do not photosynthesis. Okay, this is the classification. Why Rafflesia is so important? Because one of the plants protected by the government from extinction is Rafflesia. This plant is endemic to Sumatra, and there are only 33 species in the world. In addition to being designated as a national plant flower, this charismatic giant flower, which originates from Bengkulu, is included in the list of protected plants under government regulation. And IUCN red list with the status of conservation for being endangered species.
Beneficial to some insect species. There are 3 insects found, namely Drosophila colorata, Chrysomia megacephala, Sarcophaga haemorrhoides, which actively visit Rafflesia flowers. And food research for large mammals is some large mammals are interested in flower buds of Rafflesia species. Mammals make Rafflesia flower buds as food. Next. Factors causing a decline in the population of Rafflesia. 1. Habitat loss, such as deforestation and land use change leading to the loss of Rafflesia's natural habitat. Clearing forests for agriculture, infrastructure development, and urban expansion reduce the necessary living space for this plant. 2. Climate change. Change in weather patterns and climate change can affect the reproductive success and growth of Rafflesia. Unusual temperature fluctuation and rainfall patterns can disrupt the plant's life cycle. 3. Illegal hunting. Some Rafflesia species have aesthetic and cultural value, making them vulnerable to illegal hunting for the pet trade or private collection. 4. Disruption of pollinators. Rafflesia plants rely on pollinators such as flies and beetles to aid in their reproduction. Larval infestation and unfavorable environmental conditions can also contribute. Disturbance to pollinator population or population damaging their habitats can reduce Rafflesia's reproductive effectiveness. 5. Excessive individuals in the same root system leading to inadequate nutrient supply. 6. Inadequate planning in tourism activities. And 7. Lack of protection. The absence of sufficient protective measures for Rafflesia make the plants susceptible to the aforementioned threat. Next.
Okay, so there is a few conservation strategies that already tested in Indonesia, included an exit conservation of Rafflesia. These conservation are done in Bogor Botanical Garden, or BBG, that was established in 1870 and is the oldest botanical garden in Southeast Asia. These facilities are located in West Java that recorded a milestone on Thursday when it found that Rafflesia, the species is Rafflesia arnoldi, that had bloomed in the iconic gardens far from its native forest habitat in Sumatra. Next. In that facilities, there are several efforts made on Rafflesia conservation that include seed inoculation, in vitro culture, and grafting. Next. Okay, for the first effort is in vitro culture. So, okay, the procedure is sorry. Next. Okay, so the first sorry, come back, please. Okay, so for the in vitro culture, the procedure is by putting the plant parts of Rafflesia into a culture medium that contains sufficient nutrients to support the life of the plant cell or tissues. There's some part that already tested on this method. The whole Rafflesia migerni flower bud explant, and then there's a root explant culture of Rafflesia padma that already infected. The third is Rafflesia arnoldi seed culture, and the fourth are Rafflesia tetrastigma symbiotic culture. The result is the most of the tests showed there's no growth response in Rafflesia cells or tissues that cultured on culture media, and there's only one result that show an increase in the size of the tissue, especially for the flower bud. There's the growth diameter from the start that is larger than before. And these efforts are still being studied to get better results. Next. Okay, the second effort is seed inoculation. In this effort, the procedures are by inserting or sprinkling flower seeds into the slash part of the host tree or trunk root. Yeah, the part is the trunk or the roots. Both the host tree, the Tetrastigma sp, in the native habitat and in the Bogor Botanical Garden. So you can see this is the procedure, and by the result is this method, the inoculation on the host tree, both in the native habitat or in the Bogor Botanical Gardens, did not show any seed germination. But the condition of this Rafflesia seeds looks fresh and full. It's the hypothesis is why it still looks good is related to the physiologies of the Rafflesia itself. Next. Okay, the third effort is grafting. The procedure is by inserting or connecting the parts of one plant to another plant in such way that there is a compound between the two plants that are connected and are permanent. The grafting method chosen is root grafting, either root pieces or the whole roots. In this case, the Rafflesia used the root part as the method. And there's two methods in grafting that use it. The first is Wiener grafting method, as you can see on the upper picture, and the second one is live grafting method. For the Wiener grafting method, it involves the cutting the tip of the scion into a V shape and then inserting it into the tetrastigma root, which acts as the rootstock. In this method is chosen if both sides of the roots are at the same size. But for the cleft grafting method, by inserting the scion into the rootstock root incision. This method are chosen if the size of the roots are different. And the result by this method is since 2004 till 2060, at least 10 rafflesias flowers are successfully bloomed. Next. I will pass it to the next presenter.
Rafflesia species are entirely dependent on their host, Tetrastigma species, by receiving water and nutrients from it, as the parasitic plants have photosynthetic capacities. Ultimately, studies of Rafflesia have been carried out since 2004, with emphasis on Rafflesia patma. Since 2004, attempts to grow Rafflesia patma have been conducted in the ex situ conservation area of the Bogor Botanical Gardens. In order to perform ex situ conservations, studies on in vivo and in vitro cultivation and conventional grafting methods were tested. After the first registered ex situ of the two methods, grafting, cut, and veneer, on the house plant Tetrastigma cariosum, has become successful for the growing of Rafflesia patma in the Bogor Botanical Garden. The flowering has occurred 10 times since 2010, 8 female and 2 male flowers, where all 10 flowers came from 3 different hosts, and only 2 times produced male Rafflesia patma flowers. The grafting method is a short way to the Rafflesia reproduction. Thus, when major seeds are difficult to obtain or these are not available, the grafting method has become a successful alternative way for its cultivation. The grafting method gives new hope for the establishment of efforts of ex situ conservation of Rafflesia. However, the next step is much more difficult. The unisexual Rafflesia patma flowers are either male or female. The female flowers could not reach further males, no pollination has been registered. The successful effort to grow Rafflesia patma in ex situ cultivation is a small step leading towards a larger scenario of population establishment. Experience obtained in this initial successful attempt provides the basis for more complex work. It implies carrying out both human-induced and natural pollination experiments as an effort to establish a viable population. Moreover, most of the localities of Rafflesia patma are in West Java, but it is also present in Central Java and non-historically from Sumatra. According to separate studies by Moglia and Ais, these regions are threatened by deforestation for palm oil production and natural disasters such as tsunamis. These authors note that buds of Rafflesia patma are used in local medicinal practices and are therefore subject to intense harvesting. Next slide.
Okay, the next topic about the importance of educating and involving the community in conservation. Increasing public understanding of Rafflesia is important because Rafflesia is one of the unique and fascinating wonders of nature. Rafflesia is a genus of parsley flower consisting of several species, including Rafflesia arnoldi, which is known as the largest flower in the world. Efforts to involve community in conservation should begin with knowing how much they know and understand about the resources and their importance to them. This is important because the perception and attitude of those directly involved in the utilization of various biological resources determine the success of certain conservation activities. Next. So this is the reason why it is important to increase public understanding of Rafflesia. So the first one is natural conservation. We know that Rafflesia is endangered due to habitat loss and illegal hunting. By increasing people understanding of the GDF importance of Rafflesia in the ecosystem, they will be more likely to support conservation efforts to protect the species and the environment in which it lives. Second one is education and research. Rafflesia has unique properties, including its depends on a host to grow and thrive. With a better understanding of Rafflesia, communities can support wider research and education effort on flora and fauna in the local environment. The third is ecotourism. Rafflesia can be a strong ecotourism attraction due to its appeal as the world's largest flower and unique use as a parasite. Improving public understanding of Rafflesia will help develop the potential of ecotourism that is sustainable and profitable for local community. The fourth one is environmental awareness. Rafflesia is an important indicator of environmental health due to its dependence on a healthy natural habitat. If Rafflesia population are declining, it can be a sign of larger environmental problems. A better understanding of Rafflesia can raise public awareness about the need to maintain a balanced ecosystem. The last one about economic potential. Increased understanding of Rafflesia can also open up local economic opportunities, such as the development of Rafflesia, best crafts of souvenirs, or Rafflesia-related tourism service that can increase income of surrounding communities.
Conclusion of this discussion, the dedicated action taken to preserve the Rafflesia species through the exit to conservation at the Bogor Botanical Gardens play a significant role in achieving sustainable development goals. By carefully nurturing and protecting these plants, the garden have maintained a healthy environment and diverse ecosystem. This also helps people learn more about nature and the importance of protecting the different species. In short, the efforts to protect Rafflesia at Bogor Botanical Garden contribute to a better and more sustainable future for everyone. Next. So this is our reference and this discussion. Thank you for your attention. I will pass it back to the moderator. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Okay, thank you very much, UNSOED Group 1. Now the paper is open for discussion. Are there any question or comment?
I have a question.
Oh, please.
How long does it take for Rafflesia to flower?
Do you understand his question?
Yeah, we do. Can you keep? Okay. So, for how long the Rafflesia will grow in the native habitat? It's about 4 years till start from the seeds till it blooms. It takes about 4 years.
Thank you.
Okay, I have a question. So I'm afraid I missed something, but you mentioned that the Rafflesia use smells like rotten flesh to attract insects. I thought that the object to attract insect is pollination. Is my understanding right?
Excuse me, sir. Can you repeat the question, please?
Yeah. Okay. So in the presentation, it is told that Rafflesia attract insects by using the smells like rotten flesh. Then I thought that the object of the insect attraction is pollination. So is my understanding right?
Yes, that's right.
Okay.
That's true.
So,
yeah.
Okay. So, if so, why the Rafflesia uses smells like rotten flesh? So I think there are many insects. So why the Rafflesia select the insects which likes the smells like rotten flesh? Are there any reason?
Okay, thank you for the question. As long as I know, there is a few species that only attracted on rotten smells like this, like the fly or something like that. So that's some selective selective preference that because the Rafflesia arnoldismers are like rotten flies, so the insect that attracted to the rotten scent are that only a species that will come to the Rafflesia.
So at first I thought that in the niche of the Rafflesia, only the insects which like the rotten flesh are available for the Rafflesia. But are there more insects exist in the niche of the Rafflesia? Yeah, so my question is, why Rafflesia select such insects? To avoid the, I don't know how to say, other flowers also attract insects for the pollination. But I think the insects which like the rotten fleshes are very specific to Rafflesia. So they avoid the competence with other plants in the meaning of the pollination. Yeah, I'm not sure. So yeah, if you know.
I'm sorry, this we are not really know about that, but maybe we will looking for the answer.
Yeah. Okay. Thank you very much.
Thank you.
Okay, are there any questions or comments?
Question? Hello?
Oh, please.
May I have question?
Please.
Okay.
Please.
From me, yeah. Just now you inform us about the intensive harvesting, yeah. So, can you explain again what the purpose of this one? I mean, why people, society harvest this one? This is the first question, and then maybe after that I will ask again.
Thank you, Tim Sensei, for the question. So that we already said for the purpose of the people to collect this flower is for a traditional medicine purpose. Yeah. So our people use this as medicine, traditional medicine, but we are not really sure what kind of medicine that they are trying to make.
Okay. Thank you very much. So therefore, my next question is, I think we can learn from the edibleness sweetlet, edibleness sweetlet, or we call this burung walet, sarang burung walet. Yeah. So I think previously that this sweetlet is endangered, and sometimes and then of course the world also said that it is illegal trading for this nest. Yeah. But after people in Indonesia or also in Malaysia, or also in Filipina involved in order to establishing the sweetlet house. So starting from 2009, so this is the list of the endangered species because no abandoned, and also have some involved of this bird for pest what is it, pest problem, yeah, so they can pest control, yeah, so because this bird also will eat insect that it is also pest. So therefore if you see that there's kind of the economic thing, so I think human being also can interfere for this kind of the Rafflesia. So what do you think? I think it is possible for Rafflesia, and one day Rafflesia could be like edible nice sweetlet. What do you think about this one in the future?
Okay, thank you, sir. So based on this, our research before that, maybe in the future, if the methods to how do we say, to make the individual as much as we can in this conservation,
to propagation, yeah.
To propagation the species, maybe in the future we can use Rafflesia as consume it, but for this time there's still no effective way to really increase the total individuals
for this species. So maybe we still need about 5 or maybe 10 years later to make this rafflesia as our consumption, something like that.
So can we conclude that there's some possibility, yeah, one day? Okay,
Yeah.
thank you very much.
Thank you for the question.
Okay, are there any question or comment? Okay, if no, let's move to the next presentation. Thank you very much, UNSOED Group 1. Okay, next presenter is KGU Group 4. So if we are ready, please start your presentation.
Hey, hello everyone. We are Group 4 from KGU. Today, we would like to discuss correlation between human health and biodiversity. The title is Applying the Wisdom of the Local Forest to Staying Healthy. Let me first introduce our member, Aoi Otake, Rina Shibatani, Ayana Oshiro, Suzuka Ogura, Takeyuki Sasahara, Takuya Fukutani, and myself, Konomi Takebe. This is the outline. Our presentation is divided into 5 parts. We will start with the discussion on biomimicry and biodiversity. This is outline. Our presentation is divided into 5 parts. We will start with the discussion on biomimicry and biodiversity, and then move on to explain the various use of natural wisdom with a few examples. And finally, we make some suggestions for the future. In 2015, the United Nations established 17 goals, Sustainable Development Goals, SDGs. The third goal represents the well-being of human beings, while the 14th and 15th are for biodiversity. Initially, they seem to be completely independent and have no relationship with each other. However, they do co-create our bright future. In addition to the global goals, the World Health Organization, WHO, also pays attention to the close relationship between human mental and physical conditions and the diverse range of species. Furthermore, through conserving the Satoyama environment, which represent the harmonious balance between nature and human activities, we have come to realize that diversity of living things can have profound impact on our health. Various creatures in the Satoyama environment have shown positive effect on our well-being. Today, by deepening this strong connection, we would like to clarify the special gift that Mother Nature gave us, and we would like to share with you how biodiversity influenced human health as well as plants and animals.
Humans have been applying the wisdom of nature in various fields, from medicine to engineering. Particularly, humans imitate the engineering of the natural world and create innovative solutions. This is called biomimicry, and through this, we have made great progress in science and technology. For example, the shape of a streamline of the Shinkansen is inspired by a beak of birds, such as a platypus, and reduces air resistance. In addition, biomimicry is also used for the development of swimsuits with properties similar to shark skin. Also, injection needles are inspired by mosquito masks. In addition to biomimicry, you can get direct benefit from natural resources and living things. Functional markers that are essential to our health, such as valine, tyrosine, and phenylalanine, cannot be synthesized in the body, but with the progress of molecular biology, the search for functional compounds is becoming more and more active. Each species has unique wisdom and characteristics that can be used for human health. Therefore, how to draw out the power of biodiversity is getting more and more important. In order to use natural resources and organisms for health promotion, it is important to research and investigate how to use them. For example, by extracting specific plants and herbs from biodiversity and using their fragrances and health promotion can be expected. People have used a lot of biological resources so far. Even though species are said to be good for our health, most of them do not directly affect our bodies. It is not living things themselves, but chemical compounds in their systems that have direct impacts on us, and we can develop our health thanks to them. We have classified such chemicals based on how they affect us as follows. Substitute for harmful substances, functional substances, defensive molecules, and healing function. The first category, substitute for harmful substances, is that we can replace harmful chemicals with safer, natural origin substances. The next class, supplements for nutrition, is that the molecules themselves have some benefit in making us healthy. Nowadays, more and more people are using supplements, and that is an example of the second way of their usage. Another genre, defensive molecules, seems to have more things to do with medicine much more directly. Today, a variety of medicine is used to cure various diseases. Humans have used natural substances for this for a long time too. Even today, there is a database called knapsack, which summarizes the function of the metabolites that can work as if they were medicine. As most medicines are inspired by creatures, this category is also inevitable. The last way to use such components is healing function, such as herbs or aromas. There are significant effects that perfume from nature can heal people, which can make people healthier. It suggests that this category of usage also plays quite an important role in our health. With this various use, many more species tell us a lot.
Many kinds of herbs planted in wetlands like it are used to keep our health. Lush japonica called Nurude is one of the greatest examples that can be used for maintaining our health. There are some pictures of Nurude. Nurude is categorized as one of sumac families. Generally speaking, this family has toxicity, so we cannot touch it barehanded. However, since Nurude doesn't have toxicity, so we're able to touch this herb. Surprisingly, Nurude contributes to three directions: defensive molecules, functional substances, and healing functions. Let me introduce three ways of Nurude's considerable effects on people's health. First, Nurude is thought to exert an antimicrobial effect. When an aphid parasitizes Nurude, it forms a tissue called an insect bump as a defensive response. After the aphids hatch, the insect bumps contain high levels of tannin, which has antibacterial properties. When injured, the liquid from Nurude insect bump can be applied to wounds inhibited the growth of bacteria. Second, it can be used as a hair dye. The aforementioned insect bumps are black and contain tannin. which was used as a coloring agent for dyeing gray hair in old days. We believe that it is possible to extract pigment components and use it as a naturally occurring dye even today. Finally, Neruda has a potential to be used for fruit products. In the fall, Neruda secretes a substance called calcium malate on the surface of the berries. And when dried, when dried, this salt can be extracted. This white substance around fruits is calcium malate. And this substance has a salty taste when you lick it, but it is not a salt. We wondered it could be used as an alternative to salt in food for patients need diet without salt. Since calcium malate is harmless to human consumption, it can use as a salt flavor is highly possible. Plants native to wetlands like it have the potential to make a significant contribution to the improvement of people's health because of their various effects.
We now move on to the second example, Nezumisashi, as natural resin. Its scientific name is Junipers rigida. It is evergreen conifers which grow in hilly areas, rocky areas, and ridges. This plant's leaves are very sharp. When spring comes, these trees flowers bloom and bear fruit later. In the autumn, a few years later, the fruit's color turns black and dark purple. In Japan, it grows in the southern west part of the country. The adult Nezumisashi hardly withers. However, it grows quite slowly, so it hard becomes an adult tree. Actually, 30 prefectures have registered it as a semi-endangered species. Even though the tree is useful, we had better think of the balance between its use and the impact on the ecosystem. By using Nezumisashi under control, we can make the most of it. The adult itself is mainly used for construction material such as Buddhist tools, chopsticks, and so on. The plant has positive effects on our life. In addition, the fruit is dried and used as bath salt, medical liquors, and perfumes. When you use it as a bath salt, it smells good and you feel better. This is nothing but the healing function itself. As an effect, the active ingredient includes essential oils, beta-penic acid, and so on. Its oil promotes the discharge of waste products in the body and is expected to have the effect of relieving tension. As with other effects, it is effective for rheumatism, neuralgia, and cold. Now, a lot of people have many stresses. We hope that you can use them as an opportunity to enrich our lives even a little.
Now, as a third example of nature's blessing, we introduce Lindera umbrella. In Japan, Lindera umbrella is famous for being used as a wooden pick for luxury Japanese suites. The feature of this wooden pick is having elasticity, high strength, and resistance to breakage. In addition, the fragrance is so good as to be used as a perfume and cosmetics. We started to learn that Lindera umbrella is also used as a tea leaves after participating in this certain conservation activity. We drank Lindera umbrella tea for the first time, and it was tasty. I felt like it had some positive effect just like other herbal teas. In fact, this tea has 3 main ingredients that contribute to health improvement. The ingredients are cineol, linalool, and caffeine. Cineol is often used in throat lozenges. You can expect the effect of soothing inflammation in the throat, removing phlegm, and adjusting immunity. And it is effective as a prevention of cold. In addition to its calming effect, linalool also has an antibacterial effect against dental problems such as tooth decay, suggesting this is not just healing function, but also the functional molecule and defensive molecule. This ingredient contributes to oral health. Caffeine is a component that helps blood flow smoothly and keeps the body warm. By warming the body, the immune system is enhanced. In this way, Lindera umbellata has components that contribute to health, and people can maintain their health by extracting these components and drinking them as tea.
So let us sum up our discussion. Humans have been using biological wisdom in many fields, especially in medicine field. And so we can utilize natural wisdom to human health, and thanks to the molecular advance of molecular biology, we now have great access to such intelligence at the molecular level. So by harnessing and applying them in many ways into medicine, including substitute for harmful substances, functional substances, defensive molecules, and healing functions, we can improve the whole health conditions better, which can lead to the sustainable society, one goal of SDGs. However, recently, concerning trend represents the source of such intelligence has been lost owing to human activities, and the number of the number of the species in the West has been increased year by year. So this means the loss of intelligence. So, but the only way to conserve such intelligence is to let me fix it. To stay in the harmony with natural environment or natural resources. So by doing so, we can have a brighter future by staying in harmony with such natural resources. We should respect Mother Nature. So, and that will lead to the better future by using the greatest gift that nature gives us as biodiversity, and we can have both sound relationships with human health and also the natural environment. So that's why we should entertain a profound respect for Mother Nature, and by doing so, we can build a sustainable relationship with them. So thank you for listening.
Okay, thank you very much, group of KGU. Before starting the discussion, I have one comment. You used the term creature, but the recent use of creature, the plants are not included. So the creature is not the adequate term. Okay, it's a minor comment. So are there any question or comment?
I have a question.
Okay, please.
What is the most important thing about this presentation?
So the key terms or the most important things of this presentation is that the biodiversity has directly benefit for our health, for us to stay healthy.
Thank you.
Okay, are there any question or comment? Okay, if we now let's move to the next session, the presentation. Thank you very much, group of KGU. Okay, the next presentation as a final presentation. So the presenter is UiTM Group 4. Okay, if we are ready, please start.
Can you hear my voice?
Yes.
Okay, so I will proceed, but wait for a minute. So sorry. Okay, so hi and a very good morning. We are from Group 4 from UiTM Shah Alam. I'm Hanidana bint Abdul Aziz.
Arifah binti Muslim.
Hi, my name is Wana Udina binti Rosman.
Hi everyone, my name is Wan Hazwani.
Hello, my name is Shatika, and Group for UiTM will be presenting biodiversity conservation of the.
Honey, next slide. Okay, before we start our presentation, I would like to explain about SDG or Sustainable Development Goals. SDG are a universal action to end poverty, protect the planet, and ensure that by 2030, all humans and creatures enjoy peace and prosperity, which are the 2030 agenda. As stated in the slide, there are 17 SDGs integrated, which are no poverty, climate action, good health and well-being, and etc. Where the actions from these areas affects others. Therefore, the development of SDG must balance social, economic, and environmental sustainability of what their goals are. Some of the examples of SDG actions done in Malaysia are the establishment of Animal Welfare Act 2015 and the enhancement of quality of urban areas, for instance, reduction of carbon emission. I will pass it to my friend.
Okay, so zebra, or the scientific name Equus quagga, is classified in the IUCN Red List as a near threatened species in which the numbers are declining. Zebra is not a native species in Malaysia. However, the concern of its declining population is still quite alarming, as the hunting for plain zebra still occurs during these days and time. Overhunting of zebras because of their meat and their unique coat poses a serious risk to its population, explaining the decrease in number of its species. It needs to be understood that extinction of this species and any flora and fauna happen not only due to climate change but also illegal trading of endangered species. Other than the efforts made to conserve the biodiversity of these species, such as the in situ and ex situ conservation, modern science fields, such as biotechnology, can be implemented, in which molecular approach falls under the biotechnology category. Biotechnology is a powerful modern tool that has recently been increasingly utilized to acquire this knowledge for conservation, evaluation, and application of biodiversity. Challenges that require basic knowledge of evolution and interactions among millions of organisms is made possible by the use of technology in the areas of genomics and metabolomics that are advantageous for the identification and environmental sectors. In this presentation, 5 molecular approaches such as DNA barcoding, in vitro fertilization, molecular markers, RFLP, RAPD-PCR, and eDNA will be discussed further by the members of the group. So, what is DNA barcoding? So, DNA barcoding is a molecular technique that utilizes a short, standardized region of DNA to identify and classify a species. It involves comparing the DNA barcode of an unknown organism with a reference database called barcode database to determine its species identity. DNA barcoding can assist in species identification, monitoring, and detecting illegal trade. So how does barcoding work in identifying species? So this figure summarizes the DNA barcoding framework. So the proposed system of DNA barcoding for species identification by the scientific community in 2003 is just like scanning of the DNA of any organism with the same features, just like at the supermarket scanner. By using DNA barcoding, processed goods can now be identified and their origin can be tracked, preventing illegal trading. So the pipeline of barcode production comprises of 4 steps. The specimens are obtained or stored in gene banks, national parks, and zoological gardens. Secondly is the laboratory analysis, which was done in molecular biology lab. where extraction of DNA was obtained from the specimen of any interested organism. The obtained PCR data will be represented as nucleotides and saved in the database for further analysis. Next, in the database, unknown specimens obtained from previous methodology, which is the lab analysis, will be assigned to recognized species. The barcode of life database and international nucleotide sequence database, GenBank and BO, are the main barcode databases. Lastly, the data specimens will be analyzed and identified via closest matching reference record in the database. So DNA barcoding have few applications. So another one of the application is the identification of species, rediscovering species, and to discover differences in diagnostic. So DNA barcoding includes rediscovering of species. For example, that were thought to be lost, as barcoding allows some flora and fauna species to be rediscovered. This is proven by the rediscovery of predatory water beetle species after it was thought to be extinct for 26 years. DNA barcoding also aids in discovering the differences in the diagnostic, as this method helps taxonomists to distinguish species and prevents misclassification of species into other groups by taxonomists. It is important to implement DNA barcoding to prevent the loss of endangered and vulnerable species, whether it is flora or fauna. DNA barcoding benefits could aid in the identification of processed illegal trade so it can be traced back to the original place. It is recommended to implement DNA barcoding globally to conserve any endangered species to conserve the biodiversity of the flora and fauna. So next person.
Another molecular conservation method that can be used for conservation of species is in vitro fertilization, or IVF. Honey, next. Honey, next time. IVF, or in vitro fertilization, is a commonly used molecular conservation procedure where, by definition, it is the process of collecting eggs from a female ovary and fertilizing it with a male sperm in a laboratory. IVF is considered to be part of assisted reproductive technologies, ARTS, aside from ICSI, which are intracytoplasmic sperm injection, which both are used for conservation of endangered species. Next slide. There are three main importance of IVF, which are stated in the slide. First is to overcome the psychological problems which may lead to non-productive species by traditional methods. Therefore, IVF aids in producing offspring from these elite animals. On top of that, preservation of valuable genetics is possible with the assistance of IVF. This is to produce offspring with favorable genetics. Lastly, IVF helps in shortening generational interval, therefore increasing propagation of premium livestock and increasing its population. Next slide. Now I am going to talk about the methods of IVF. There are generally 5 steps to IVF, starting with collection, where donors with superior genetics undergo oocyte collection using an ultrasound-guided needle. The collected oocyte will then undergo a maturation process in the lab that will take about 21 to 24 hours. After the oocyte has matured, it is fertilized by introducing it to sperm and co-incubated overnight, or approximately 16 to 20 hours. Following fertilization, the embryos are then cultured for 5 to 7 days. Hence, the final step of IVF is given to 2 choices, which one, to transfer the embryo to a recipient animal to grow and develop, or to freeze the embryo for transfer at a later date by using cryopreservation. IVF on zebras aren't used much in Malaysia. However, Malaysia integrates this method on cattle such as cows, buffalo, and bison. Next. Therefore, as I said before, IVF or IVP in vitro production is not commonly used on zebras in Malaysia. However, it has been used in other countries. Malaysia just use IVF for cattle such as bison and cows. That's the end of my presentation. I'm going to give it to my friend.
Next. RFLP stands for restriction fragment length polymorphism. It is a versatile technique that can be applied to analyze genetic variation in the DNA of different species. By using restriction enzyme to cut the DNA into fragments and separating them through gel electrophoresis, RFLP can help to identify and compare genetic differences within and between zebra populations. This technique can be valuable in studying the genetic diversity, population structure, and relatedness among different zebra individuals or population, which can aid in conservation effort and understanding the evolutionary relationship within the species. Next. Why use RFLP in zebra? Firstly, it's genetic diversity assessment. RFLP analysis help to assess the genetic diversity within zebra populations. It provides insight into the variation in the DNA sequence, allowing researchers to understand the genetic structure and relatedness of individuals within and between populations. Secondly, population differentiation. RFLP can be used to study the genetic differentiation between zebra population. By analyzing the pattern of DNA fragments, researchers can determine the genetic similarities and differences among different populations. Lastly, marker identification. RFLP can identify genetic markers associated with specific traits or disease in zebra. This marker can be utilized for various purposes, such as selective breeding program, studying genetic disorder, or identifying individual carrying specific genetic trait of interest. Next. Next, we go to the method of RFLP. The first step is sample collection. Collect a DNA sample from the zebra. This could be from blood, hair, and skin. Second, DNA extraction. Isolate the DNA from the cell in the collected sample using a DNA extraction procedure. Third, restriction enzyme digestion. The isolated DNA is then cut into the fragment using restriction enzyme. This enzyme recognizes specific sequence in the DNA and cut the DNA at those points. For gel electrophoresis, the DNA fragment are separated by using gel electrophoresis under the influence of an electric field. Smaller fragments move through the gel faster than larger ones. This creates a pattern of bands, each representing DNA fragments of a specific length. Fifth, visualization. The DNA fragments in the gel are made visible, often using a dye that binds to DNA and fluorescent under ultraviolet light. This allows the pattern of band to be seen and photographed. And lastly, analysis. The pattern of band is unique to the individual zebra, and it can be compared with pattern from other zebra to answer various genetic questions. For example, it can be used for parentage testing, population genetic studies, or identifying genetic diversity. I will give to my next presenter.
Okay, next technique is RAPD-PCR. Next slide. Okay, what is RAPD-PCR? RAPD-PCR is known as random amplification of polymorphic DNA, RAPD, involved using short, random primers to amplify random sections of genome. This technique is utilized to evaluate genetic variation and determine the population structure within species. The purpose of use RAPD-PCR in zebra is to study and understand genetic diversity, phylogenetic relationship, and also population genetics. This technique allows researchers to analyze the DNA of different zebra populations and individuals to determine the level of genetic variation within and between populations. So how does it work? RAPD-PCR works by amplifying the random regions of DNA, result in the generation of unique bending patterns. These binding patterns are reflective of the difference in DNA sequence among individual or population being studied. The technique utilizes short random primers that bind to multiple locations in the genome. During PCR amplification, this primer initiates the synthesis of new DNA strands, resulting in the amplification of random DNA fragments. resulting in the DNA fragments, result in amplified fragments, then separated and visualized. Often used in gel electrophoresis, which produce distinct bending patterns. This variation in these bending patterns indicate the difference in the DNA sequence between individual or population. Next slide. Okay. There are 5 steps. Short random primers are chosen for the PCR amplification. These primers have random sequence and can bind to multiple locations in the genome. Next method is DNA extraction. The DNA of sample being studied, for example, zebra or zebra population is extracted from their cell or tissues. The third method is PCR amplification. The extracted DNA is mixed with the selected random primers, DNA polymerase enzyme, and nucleotides. The PCR reaction is carried out through a series of temperature cycles, which allows for the amplification of random regions of the DNA. The random primers bind to their complementary sequence in the DNA template, and DNA polymers extend the primers, result in the synthesis of the new DNA strand. The fourth method is gel electrophoresis. The amplified DNA fragments are separated and visualized using gel electrophoresis. The PCR products are loaded onto gel matrix and subjected to an electric current, causing the DNA fragment to migrate through the gel. The smaller fragments move faster and travel faster than the larger one. This suppression results in distinct bending patterns which represent the amplified DNA fragment. Lastly is data analysis. The bending patterns are obtained from gel electrophoresis, analyzed to determine the genetic variation and population structure. The presence or absence of specific bands in different individuals or population is compared to identify the uh, genetic, genetic differences or similarities. Statistically, statistical analysis and comparative diversity, phylogenetic relationship, and also population genetics. Next, I will pass to the next presenter.
Another method that can be used is quantitative study of eDNA and application of next generation sequencing. Next. Okay, so before we go into further detail, let us understand what is eDNA. So eDNA is mitochondrial or nuclear DNA released by animals into environment, and it is important in providing a novel source for researchers and conservationists to observe and record alien and invasive species. Next. Okay, now let us have a look on how quantitative study of eDNA help in conservation of zebra. So quantitative study of eDNA measure a species presence absence in natural ecosystems and its relative abundance. It has the ability to record number of species present, number of individuals living within any given environment, which able to change ecological inquiries to change from species richness to species diversity. Next. Okay. Other than that, it also provides advanced data for biodiversity and ecosystem monitoring that allow track changes in ecosystem over time and understanding of ecosystem health. Thus, we can identify individual zebra from a far understanding zebra subpopulation. Next. So there are 2 suitable methods for eDNA extraction. The first one is filtration. Next. And the second one is precipitation. Next. Okay, and some of the next generation sequencing used in conservation of zebra is metabarcoding. So metabarcoding is a process of analyzing numerous species at the same time. And this method, we can identify the presence of zebra species in a place, for example, in savanna. And by using this method, we can identify the difference between species through one analysis process and clarify the taxonomic composition of animal in multiple places. And in this method, primers are designed to identify a similar portion of DNA across all species in a group. So by using meta-barcoding, we can identify endangered zebra species. Next. Okay, we can also use high throughput sequencing, which is a technology that allow analysis of million sequences at the same time. And it is able to measure species richness and composition, and it is very useful in long-term monitoring and paleoecological research. So overall, these two methods provide information such as animals' genetic diversity, life histories, and disease resistance. And this information will help us to determine a better strategy to conserve the zebra. As example, we can plan a strategy on how to reduce certain disease in zebra so that their number will not decrease. Next, I will pass it to the next presenter.
Okay, so the effort to conserve species on Malaysia is the first one, establishment and maintenance of protected areas such as national parks and wildlife reserve, where native species can thrive undisturbed species. Next is Malaysia emphasize anti-poaching measures to combat illegal hunting and trade of endangered species, imposing strict penalties for offenders. Next is engaging local communities in conservation efforts is also crucial as it fosters understanding and supports for protecting wildlife. By integrating these approaches, Malaysia aims to safeguard its precious species and contribute to global conservation endeavors. Next, I will pass to the next presenter.
Conclusion of SDG is it can be said that sustainable development brings out stability in the requirement of the environment. It's made the resources available for use for the future generation, and sustainable development is an amazing way to conserve the resources provided by nature, and this can be achieved by using eco-friendly resources and technologies that will have no serious impact on the environment. And lastly, the resources that are available to us should be used in the sustainable manner so that they do not become extinct for the usage of future generation. Next.
In conclusion, it is very important for us to sustain the biodiversity because biodiversity is very important for the processes that support all life on Earth. And wide range of animals, plants, and microorganisms are important to provide healthy ecosystem. That's all from us. Thank you.
Okay, thank you very much. So now the paper is open for discussion. Are there any question or comment?
I have a question.
Okay, please.
What made you decide on this theme?
I'm sorry, what was your question again?
What made you decide on this theme?
You mean that why the zebra is selected for the presentation? Is it right? I think his question is why you selected zebra for this presentation. Is it right?
Okay, wait a minute. Can you give us a minute?
Okay, I try to answer the question. So, actually, we went to the Malacca Zoo for to complete this task to choose the molecular to choose this method to prepare this slide. So actually we randomly pick it because during our visit to Malacca Zoo, we can only see I think around 2 to 3 zebra at the Malacca Zoo, and we try to find out about more information about the zebra, and we found out that it is endangered species. So we think that it is important for us to sustain the biodiversity of the zebra. And I also found out that zebra is very important in Africa because it will maintain the grassland at the savanna. I hope that answer your question.
So is it okay, KGU?
Thank you.
Okay, so I have a question about the next generation sequencers. So the figure shows a bridge PCR and the synthesis, the sequencing by synthesis. So I think it is a Illumina system. Illumina system generates a short read. But recently, different techniques for the sequencing techniques have been developed, so such as nanopore sequencers. So the nanopore sequencers generate long read. So I think the length of the read may be related to the I don't know how to say, the efficiency to identify the species. Of course, long read, the accuracy and the quality of the long read is worse than the short read. But how do you, today you selected the Illumina system, so how do you think the which sequencer is best for the analysis? Please.
Are you asking which methods is the best for this data analysis for
Yeah,
zebra?
so which next generation sequencers are best for the so for example the species identification or something like that?
So far, based on my readings, I found out that meta barcoding could be the best method because by using meta barcoding, we are able to identify each single. Sorry.
So I'm asking about the next generation sequencers. Could you please show the the explanation figure of the next generation sequencers? Maybe, yeah. So this slide shows a bridge PCR and synthesis by sequencing. So I think it is a system of the Illumina sequencers, but there are other methods such as nanopore sequencers. So Illumina sequencers generate short read, but nanopore sequencers generate long read, and the quality of the long read is worse than the short read, but the length of the sequences, read sequences, may be related to the effect, the accuracy to identify the species. So my question is, which sequencers are better to identify the species or something like that? It is my question.
From what I understand, what you are trying to ask is which sequence, which is either long sequence or short sequence, next generation sequencing is used.
There are several,
right?
There are several different techniques for the next generation sequencers. So which sequencers or sequencing techniques are best for their purposes?
From our own research, the best sequencing method is probably the short sequencing method because it is easier for you to identify the differences, as some species have very little differences between each other. So, therefore, having a short sequence, a short reading, will give you a more accurate result, therefore a better next generation sequence.
Okay. Thank you very much. So are there any questions or comments? Okay. If no, thank you very much. Okay, now all the programs are finished. Thank you very much for active discussion. I really enjoyed all the presentations. And I'm glad if you have also enjoyed this workshop. We started this activity 2 years ago, so this is a third workshop. SDGs are worldwide problems, but each problem has specific issues to a country or a region. Therefore, as discussed in this workshop, such problems are treated by the country-specific or region-specific manner. But at the same time, our approach in a country or a region may be diverted to the problems of the other countries or other regions. I wish that this workshop can provide a place to share such information about SDGs among different countries or different regions. So we will continue and expand this workshop. We are now planning the online SDGs workshop of next year. So we are glad if you join the next year's workshop. Before closing the workshop, I'd like to express my thankfulness to all the participants for their contribution to this workshop. Thank you very much. Okay, let's close the workshop. Thank you very much again. So, terima kasih and sampai jumpa lagi.
Excuse me.
Thank
Excuse
you.
me.
Excuse me.
Would you like all the audience open the camera again?
Okay. He want to take a picture. Please open
Close
the
to the
camera.
camera. Okay. Can you open again the camera? From UiTM, I hope. Yeah, thank you for Professor Abe and Professor Secha.
Yeah, thank you. Thank you.
And the other professor, Romy. And so please close. It's very difficult. Yeah.
Okay, maybe a
Okay.
little. Bibin Sensei.
Yes.
So maybe I would like just on behalf of UiTM, I'm very well. Thank you very much
Yes.
for the opportunity to give to our student. Thank you very much, and see you next year.
Okay.
Okay, thank you very much.
Okay.
Arigato gozaimasu. Arigato gozaimashita.
Yeah, terima
Osawa ni
kasih.
narimashita. Thank you.
Yeah.
Okay.
Okay. So, wait moment. No, start increase camera. Okay. Please wait for 5 minutes. How about Professor Secha? I heard that one day you will come to Kansai Gakuin.
Yeah. Yes. Hopefully that our plan to bring a student from UiTM to visit Kansai Gakuen University KGU in Japan on next year February. If Sensei would like to accept us, I'm very appreciate. Thank you.
Yeah, we're looking forward to seeing you next February.
Yes. Next February. Thank you, Sensei. Thank you very much.
So this activity is very amazing for me. So this is the first time with the Kwangsei Gakuin University and also with the UTM from Malaysia.
Yeah.
And of course with the Faculty of Biology Universitas Jenderal Sudirman. So I hope Bipin Sensei, Fujimara Sensei, and the other can set up this program at least twice a year. At least, yes, because this is very important. Yeah. So on that time, maybe the student and also the lecturer can involve something like this event. Okay. So thank you very much. Arigato gozaimashita. And see you again in next event.
Yeah, okay.
Bye-bye.
Thank you very much. So I will close this. Thank you very much.
Thank you.
Okay, thank you very much and terima kasih and sampai jumpa lagi.
Okay, sampai
Sampai jumpa lagi.
Yeah.
Thank you.